So let's start, and genetics has always been of interest. The special journal we have was in the various journals, archives over the years. You can see the talk, and also it hit the general population journals like Newsweek, Time magazine, and you want to know my future? Right there, your genetics. Now, genetics in the eye has at least 90% of the genes in the human genome that's expressed in the eye. So therefore, when we're studying the genetics of the eye, we're really talking about the genetics of the body. Here's a list of the genes that affect the eye. It's amazing. You can see so many of the general syndromes that have an ophthalmology overlap, like Marfan's, Stargardt's disease, of course, for the retina. So this is our talk for me this morning. I'm going to review genetics. I'm going to talk about current gene therapy and proposed future gene therapy. On top of that, there is an elephant in the room: artificial intelligence. So here's our crash course, Genetics 101, just to refresh your memories. We can see that the gene is mostly introns, and it's the exons that we're more interested in. It's interesting that the difference from one human to another is just 2% base of 1%, tenth of a percent of the base pairs. But for a human to a chimpanzee, it's just 2%. We're very close to a chimpanzee. So here's the spread of the chromosomes. The largest ones start at number 1, and the smaller ones at the end, except for number 21 and 22. They switch over. And here's a review of the chromosome. In the center is the centromere, at the ends of the telomeres, and they're numbered as you go up for their locations. And we know about meiosis and the crossover and how this is passed on and broken down. Of course, if it occurs in the same body, it makes no difference in your proteins because all the genes and DNA are there. But if it's passed on to another person, then that change takes place, because here we have translocation of part of the chromosomes to a chromosome 4. In the same body, it makes no difference, but if it's passed on to your offspring in meiosis, it does. Let's talk about DNA, very interesting, and the RNA. The base is a 5-carbon sugar. It's a pentose. We have deoxyribose, where there's a hydrogen atom assigned to carbon 2, and if we have oxygen, then it's ribose. Attached to that is the base, and we'll talk about that to number 1, and the phosphates on number 5. So therefore, a nucleoside becomes a nucleotide when you have the phosphorus attached to it. So here, this is it. We have 2 purines, bases, adenine and guanine, and you see why they're that way, and 2 pyrimidines, thymine and cytosine. Here we are. And they're color-coded because when we do chromatography, these are the colors that we use. Now take a look at here. We could see why you cannot have 2 purines or 2 pyrimidines hook up with each other. You could see in the bases the side. It's very fat on the purines, so very fat on those bases, so they can't hook up. So therefore, a purine could hook up with a pyrimidine that's thinner, so there's more room. And of course, they are tied together with the H bonds with the nitrogen. And therefore, we then get our codons, et cetera, and our genes. When we talk about DNA and the hookup, this is what we have. It first came out in 1953. Watson and Crick formulated the structure, unbelievable with diffraction, X-ray diffraction. And they wrote about it, and then they followed a couple of months later with more of a description. And this is what we have today from Watson and Crick. Here's their talk, that the hydrogen bonds are broken, they separate, and then you could get replication. All this they figured out. Of course, they got the Nobel Prize for it. The DNA is compressed around little histones and then shoved into a chromosome in various locations. The exons are what's important. The introns got rid of it. Transcription is the elimination of the introns, and then we get the messenger RNA. And that messenger RNA, through translation, becomes an amino acid and ultimately a protein. And that takes place outside of the nucleus in the cytoplasm. And we can see it's like little plugs plugged in there. First the amino acids come and then the protein. And here's the mitochondria from ancient bacteria. And the DNA is similar to that of bacteria. It also produces the RNA and the protein. And the energy is really, in the mitochondria, adenine triphosphate and the Krebs cycle. If from back to medical school, you remember that. So here we can see the transformation of that. In the mitochondria, we have 37 genes. That's it compared to the nucleus. And here's a picture of it with its surrounding and ribosomes and the breakdown of the membranes and the energy areas. So for the mitochondria DNA, I said there's 37, but for ophthalmology, just 3 of them, just 3. Leber's hereditary optic neuropathy, Kern-Sir, and for the nuclear DNA, about 98. And hereditary is from both parents. So here's to break down the DNA, the sense, and then the opposite, the codons, the antisense, and then we have the translation into amino acids, and finally the protein. So let's talk about the open reading frame, because it's 3-letter codons based upon the DNA or the RNA. The first letter makes no sense, but then you start with the second one in threesome. So forget the F. The cow and cat ate cod, etc., etc. And then you have the end. It makes no sense if you start on the first letter. And this is the result. The amino acids, various codons, various combinations can make up the same amino acid. And it starts with a start codon, that's the start codon, AUG, methionine, and it ends with 3 of the combinations, the stop codons, and with that, the process for protein stops. But we can see how mutations can take place. We have a frame shift in which an extra base is inserted or deleted, and that changes everything, and with that, you can get various changes in the protein and diseases like sickle cell and hemolytic anemia by putting in an extra or changing the codon. And finally, the stop codon. And any single base or deletion could result in a stop codon, and that's the end of that amino acid sequence, and then you get a non-functional protein. And here's some of the associate genetic diseases, including retinitis pigmentosa. So we get rid of the junk introns, and at the end we have the encode that will continue on into the human genome and who we are. We have epigenetics. I want to talk about epi and over. This is over and above the regular genetics, and you'll see that in a minute. Darwin talked about the evolution and epigenetics, and Lamarck was based on that, because we can see that evolution is a result of some genetic changes in a population, and epigenetics is expression over and above that, It could be inherited, but it's not in the DNA, as I'll show you in a minute. We have various markers that are affected, the methyl group and the acetyl group. Therefore, we can see that parts of our body stay the same. Here we have it. Histones, our DNA is wrapped around it, and when the strand is wrapped around a histone, it cannot be touched. But epigenetic mechanisms, drugs, aging, diet, environmental, that can affect it, but it's where it's open. In the lower right, it says DNA accessible, gene is active, and that's where we have the methyl, and that is affected and can be carried out. But if it hits it on a histone, it's not altered. It's just when that tail, the DNA is accessible. It doesn't change the DNA, but it hooks onto it. So let's talk about what genetics are. It's the study of specific genes in the DNA and RNA and inheritance and mutations. But genomics is the study of all the body's genes, the whole genome sequencing, so we could have a better understanding of genetic diseases. We can see that a gene is attached in a certain area of the chromosome, as I showed you before. It's wrapped up, spiraled, and put in there. So in molecular therapeutics, we talk about genome sequencing. We want to talk about a mutant gene and DNA replacement, engineering, etc. And for the study of it, at first we were using the polymerase chain reaction. We extracted the tissue, put it in a heater to break it down, and amplified it with a cycler. And this is what I was first doing at Tony Aldavi's lab for cornea. And this is, we could see how it breaks down with the heating. It could replicate, and we could then read it according to the colors. But now it's more sophisticated. We have newer technologies, faster, higher volume, and this is at Cornell, where I'm from. And this is, at that time, a sequencer that they had. And I'm also from San Diego, and there we have Singular Genomics. And this is a brand new high-speed sequencing that leaves nothing out. Very fast analysis of the whole genome, where the DNA and the chromosomes are. It's fast readout with a small piece of tissue. And that factory, that plant is on the campus of University of California, San Diego. And here I am at the plant with the sequencer. Not a big machine, but very effective. There's home testing kits, you could read them. By the way, 23andMe is now bankrupt, out of business. Others have taken over. You can see Illumina and Singular at the bottom. Illumina is also on the campus, or near the campus of University of California, San Diego. So now we talk about the naming of genes. The Hugo gene was set up because so many researchers, when they have a gene, they put a name on it, and there was no consistency. So this organization was set up to name the genes as we knew them, 19,000 of them. Even so, they have to convince the researchers to continue the proper naming of the genes. Craig Venter, it says Boston, Massachusetts, but his lab is also adjacent to UCSD. And it's very interesting because with him, it's like a computer. You change the structure, you change the input, and you get a different output, just like with the computer. When you change the DNA software, you change the species. Just like with the computer, you change the ones and the zeros, and you get a different readout. So here we can see a computer binary code and the genetic code. You change it, you get something different. And this is the work that's being done in his lab. He has one Nobel Prize winner there, too. So let's talk about gene therapy. We have vectors, they're usually the viruses, and they're altered to carry the human DNA. And we have to manipulate the viral genome to remove disease-causing genes and insert a therapeutic one, or do something with it. So use of viruses, yeah, that's the most common. Gene delivery puts a strand of DNA in a virus because we know viruses could affect the body, and we just hope that it goes to the right place in that huge genome. Herpes simplex is actually used practically, you can see, for dystrophic epidermolysis bullosa, a cloudy cornea because of defective collagen. And this work was done at Bascom Palmer in Miami by Dr. Sabata. He removed the scarring and then put the drops of the herpes on top of it with replacement genes to replace the genetics of the collagen in the cornea, and he got clear corneas. Right there, of course, cornea is on the surface and easy to get to. I propose using the coronavirus. After all, that hits everything and it keeps changing. We can see the spikes there, and you change some of those spikes, put some proper genes in there for replacement on those spikes, that could be easily carried into the body. They go into your body anyway, so let's control it. So we can see the fusion machinery and how it penetrates and goes through the membranes and into the place that you want it. We do have to aim it at the proper place, but with the coronavirus, the whole body changes. You could change the genetics of the whole body to what you want. Here's some of the vectors. The most common is the adeno-associated virus, and number 2 is the favorite for carrying viruses or altered into the body. We can see how this is a gene delivery tool. Take out the offending part, replace it, and then with that AAV, it breaks through the membranes of the cell, travels through the cytoplasm, aiming at the nucleus, penetrates the nucleus membrane, and there you are, it's altered. A new protein. There's other methods, non-genetic. Electroporation, high-intensity electric fields, short pulses that could make the plasma membrane of various ocular tissues change. And iontophoresis, that's low voltage, continuous, and you could get the permeability of the small nuclei. It could affect us in what we do. Now, if we're going into space, like the couple did for supposed to be 1 week, it was 9 months. They're exposed to galactic cosmic radiation and solar particle events. Their DNA, the strands break, the cell cycles arrest, and the cornea, the stromal edema and epithelial thickening. This was done in mice, and it could be done in them too. And they're being examined for that upon their return, finally, after 9 months. We're talking about CRISPR. Now, Jennifer Duda was instrumental in the consideration of CRISPR. She got the Nobel Prize for it. So it's clustered regularly, interspersed, short palindromic repeats. I'll ask all of you at the end if you could repeat that. And it works with the Cas9, which is the enzyme that will break the tissue, the cutting edge enzyme for the DNA, and therefore we could replace certain sections. Again, it's the clustered regularly interspaced short palindromic repeats. And remember that a palindromic repeat is a double-stranded sequence of DNA. The order of the nucleotides are the same on each strand, but in opposite directions. So in effect, it's an antisense. And the CRISPR will break it into pieces, plasmids or viruses, and it's molecular scissors to cut it. It cuts a genome at a specific place, like this, and can replace it. That's CRISPR. Now, of course, it's controversial because if it's done with embryos, you could probably not only cure potential genomic anomalies, but also you could have social Darwinism by gene modification. You could plan for a super athlete or a brilliant genius. Well, that's what it is. Finally, I want to talk about AI. At Cornell, I was with a plant geneticist in a fellowship, and we talked about AI because they're programs, models that could really revolutionize how we think and see biology and how we could predict the genomics of plants, in her case, and humans in us. What's cool about this is all they need is the input of the DNA sequence, and they could take over after that. However, Eye magazine from the Royal College of Ophthalmology a few months ago talked about threats in academia, evaluating plagiarism and artificial intelligence with ChatGPT, and this is from an ophthalmology journal. And I had this situation at Cornell with the professors who told me that they get beautiful readouts, papers from their fellows who speak poor English, but the paper is perfect. Artificial intelligence. So what he does is sits down with that fellow and asks them questions about the paper that they wrote. And I spoke to another professor who had the same problem. I told him what the first professor does, sits down and questions the student about what was written. Here are my references, and we're really off to the future. Thank you.